ನಿಮ್ಮ ವಿಡಿಯೋ ಬರಲ್ಲ ಸರ್, ಅದು ಸ್ವಲ್ಪ ಪ್ರಾಬ್ಲಮ್ ಆಗಿದೆ.
ವಿಡಿಯೋ ಬರಲ್ಲ
ಅಂದ್ರೆ ನಿಮ್ದು ಸರ್?
ಹಾ, ಅದೇ ನಡೀತು.
ನಿಮ್ಮ ಮುಖ ಕಾಣಿಸಲ್ಲ.
ಹಾ, ಅದೇ ನಡೀತು. ಥ್ಯಾಂಕ್ ಯು. Very good morning. Very good morning.
Good morning, sir.
Good morning, sir.
First of all, I congratulate you all for your wonderful success in the MCQ examination. First IA, I think in all three subjects you have done well. And keep it up. And congratulations for the toppers and high achievers. And also I tell to those people who could not score well, try to do well and attend all the activities promptly, so that you can do well. Utilize all the time whatever is available now at home. Understood? Along with your responsibility at the house, you kindly carry out responsibility of studying also. Today, in continuation with the previous class, I will talk about receptor further. So in the previous class, I had mentioned that senses, classification of senses and all those things, and area number 3.2 in the postcentral gyrus in the cerebral cortex, carriage of information through your dorsal column tract and anterolateral system. Dorsal column tract, we have mentioned vibration, fine touch, fine gradation of the temperature, all those information are being carried through the dorsal column tract of the medial lemniscal system, whereas crude touch, pressure, temperature, which are all in crude senses, they are being carried through your anterolateral system and terminating over an area 312, that is the somatosensory area number 1, as well as there is area number 2, which is called as somatosensory area number 2, capital letter S and Roman letter 2. So you have to know that is the association area. Whatever you have learned from area number 1 is being utilized by area number 2. Like that is the situation. So now we will go on to further discussing about sensory receptors. The receptors are biological transducers which are capable of converting one form of energy into, here I have mentioned that action potential. In most of the books they have mentioned that it is action potential. To be better and to be on safer side, it is generator potential or the receptor potential in the nerve fibers. So receptors are the biological transducers which are converting various forms of energy into receptor potential or generator potential. What you can understand from transducers? Transducer is a device which is capable of converting one form of energy into another form. So that is converter. That is called as transducer. So we are in the requirement of information about internal and external environment, which has to be gathered up and to be carried through, pursued by the receptor and carried through the afferent nerve and to be converted to the appropriate part of the central nervous system. So then receptors are transducers which are capable of transforming one form of energy, and stimulus is an event or the particular form of energy that evokes a specific functional reaction in organ or receptor. So we have to know that it is an event or particular form of energy. So then what are the types of sensory receptor or sensitive? What you can understand from sensory receptor? Sensory receptor, it is a specialized nerve ending or specialized cell very sensitive for specific type of stimulus, and they are able to convert the stimulus energy into electrical energy, which is called as receptor potential. And the receptor potential, if it is sufficient in magnitude, will get transformed into action potential in the nerve which is connected to that particular receptor. Structurally speaking, receptors can be grouped into 3 categories. One is bare nerve ending, second one is capsulated nerve ending, third one is sense organ. Bare nerve endings that terminate in the periphery as bare unmyelinated endings. Then number 2, capsulated nerve endings. It is in the form of specialized capsulated structures such as connective tissue lamellae. So we will discuss about all these things later within another 2-3 slides. Then sense organs, they are the specialized cells located at the end of afferent nerve. Very commonly used terminology, bare nerve ending. What you can understand for bare nerve ending? What is meant by encapsulated nerve ending? What you can understand by sense organ? Like this question may be asked in YOs, you have to answer. What is a sense organ? Sense organ, they are otherwise called as receptors. Receptors are dendritic endings of afferent neurons that are associated with a non-neuronal cells. They are called as sense organs. Dendritic endings of the afferent neurons that are associated with non-neuronal cells, such as Pacinian corpuscle. Then, what is sensory unit? Very commonly asked question, what is sensory unit? Sensory unit, it is sensory axon and all its peripheral branches. All its peripheral branches. Let's see here. All its peripheral branches. What is receptive field? Very commonly asked. What is receptive field of a receptor? It is receptive field of a receptor of a sensory neuron. It's a particular part of the body surface area in which stimulus triggers the firing of that neuron or that receptor. It is the area of skin or equivalent from where information is gathered by one receptor. That is called as receptive field. So you can see here. So you can see here what is being what you are noticing is you are applying the stimulus for this receptor. You are seeing lesser area, but here branches are more and describing more surfacial area, as a result of which information from skin or equivalent area from where information is gathered by a single receptor is more. That is called as receptive field of a receptor. Then how you are going to classify the receptors? Receptors can be classified basing on source of stimulus, type of stimulus, and anatomical classification. How you are going to classify basing on source of stimulus? So basing on source of stimulus, it can be exteroceptors, anteroceptors, telereceptors. What you can understand from exteroceptors? Receptors which receive the stimuli from immediate surrounding outside the body. So yes, it is getting the information from the external environment. Receptors are just exposed to the external environment from where the information is gathered. Cutaneous receptor, cutaneous means skin, receptors for pain, touch and temperature. Then anteroceptors, receptors which receive stimuli from within the body. For example, chemoreceptors, chemical changes of the body fluid sensed by the receptor called as chemoreceptors. Pressure changes within the vessel, baroreceptors. Proprioceptors, length and tension, length of the muscle, yes, movement at the joint, yes. Then concentration of water, osmoreceptors, concentration of glucose in the body fluid, glucoreceptors. So they are being referred to as receptors present within the body, anteroceptors. Then telereceptors. What you can understand from telereceptor? Telemedicine. Tele means receptors that receive stimuli from the distance. For example, visual receptors, rods and cones, cochlear receptors, hair cells, olfactory receptors, smell receptors. Understood? So they are being referred to as telereceptors. So here we can classify like this. The flow chart can be given. Yes, cutaneous receptors can be divisible into touch receptors, tactile, pressure receptors, cold and warm, thermal receptors. Then pain, nociceptors. So here, touch, Meissner's corpuscles and muscle disc. Then pressure, Pacinian corpuscle. Cold, it is Crosses end organ. And warm, that is Ruffini's end organ. Pain, that is free nerve ending, nociceptor. Then second class of exteroceptors can be chemoreceptors. Taste, smell, taste, gustatory. Gustatory receptors are taste buds. Smell, olfaction. Olfactory receptors in the nose, nasal mucosa. Nasal mucosa. Telereceptors. So from the distance. So yes, stimuli from the distance. You are going to get the information from the distant object, far away from the body. Yes. So yes, vision. So vision, rods and cones in retina. Hearing, hair cells in organ of corti. So then, anteroceptor, spelling mistake, anteroceptor, E-N-T-E-R-O. Anteroceptors. Anteroceptors can be visceral receptors, visceroreceptors and proprioceptors. Proprioceptors and visceroreceptors. Visceroreceptors, here stretch receptor, baroreceptor, chemoreceptor, osmoreceptor. Stretch receptors. So yes, you can see plentifully in heart and blood vessels. Baroreceptors, blood vessels. Chemoreceptors in gastrointestinal tract, need not be in the gastrointestinal, other part also, but plentifully you can notice with a good appreciable number in the gastrointestinal tract, osmoreceptors in the urinary tract as well as brain, particularly hypothalamus. Then proprioceptors. Proprioceptors, receptors such as muscle spindle in the muscle, Golgi tendon organ in the tendon, Pacinian corpuscle in the fascia, free nerve ending and the joint, free nerve ending, pain, joints, hair cells in the vestibular apparatus. So you can see. So then, depending upon the type of the stimulus, receptors can be of following varieties. One is mechanoreceptors. Mechanoreceptors such as associated with special sense. So already we have mentioned special sense. Muscle, skin, visceral and vessels. So viscera and vessels. And thermoreceptors, skin as well as in the hypothalamus. So hypothalamus. Then chemoreceptor, olfaction, gustation. Olfaction and gustation. Nociceptor, pain receptors. Then electromagnetic receptors such as rods and cones in the retina. So yes, these receptors which respond to mechanical stimuli such as cutaneous receptors in the dermis and epidermis for cutaneous tactile sensibility. Cutaneous tactile touch sensibility. Cutaneous receptors for deep sensibility, deep pain, deep pressure. Understood? Muscle and joint receptors, and hair cells such as hair cells in the organ of cochlea, that is of auditory. So yes, for audition, hearing, and hair cells in the vestibular apparatus or vestibuloreceptors for equilibrium. Hair cells in the organ of for audition, hair cells in the vestibular apparatus for equilibrium. Then baroreceptors of carotid sinus and aortic arch for detecting the pressure changes in respect of arterial blood pressure within the vessel. Arterial blood pressure. Then thermoreceptors. What are thermoreceptors? Thermoreceptors which detect environmental temperature, cold and warm receptors. Then third variety, photoreceptor or electromagnetic receptor. Many times in YOMOs we ask this, what are the types of electromagnetic receptors? You know. So you have to tell that rods and cones are the examples. Rods and cones for the color vision of retina, which respond to light stimuli. Then third one is chemoreceptor. Already we had mentioned about this, which detect change in the chemical composition of the environment in which they are being located. Where they are being located, chemical composition of the environment is being sensed by these kinds of receptors. So yes. Here, for example, taste receptor, then olfactory receptor or gustatory receptors. If the taste or gustatory receptors, olfactory receptor or smell receptors, then osmoreceptors in the supra-optic nuclei of the thalamus, which will sense the water concentration. Aortic and carotid bodies receptors, which will detect the level of arterial partial pressure of oxygen, arterial partial pressure of carbon dioxide, and pH. Then glucoreceptors. Chemoreceptors on the surface of medulla oblongata for the detection of blood partial pressure of carbon dioxide, and chemoreceptors in the hypothalamus for detection of levels of blood glucose. We have already mentioned. So then fatty acids and amino acids. Then fifth variety under this stimulus. So nociceptors. So the receptors which respond to extremes of mechanical, thermal, and chemical stimuli which are producing pain. Nociceptor are nothing but extremes of mechanical or thermal or chemical stimuli producing pain. Then clinical classification or anatomical classification of the receptor. So remember, these are the ways of classifying the receptors, how they are being classified. So superficial receptors, deep receptors, visceral receptors. Superficial receptors which are present in the skin and the mucous membrane. Then deep receptors which are present in the muscles, tendons, joints, and subcutaneous tissue. Visceral receptors which are present in the visceral organ. Now, so we know already. So yes, number one, mechanoreceptor, epidermis and dermis, joints, tendons, ligaments and muscles, cochlea, vestibular apparatus, baroreceptor in the vessels. They are considered under mechanoreceptors. You understood? The mechanoreceptors can be present in these areas and accordingly they are being named. Thermoreceptors, warm and cool. Nociceptors for pain perception. Electromagnetic, rods and cones. Chemoreceptor, taste, smell, carotid arteries and aortic bodies. Then hypothalamus and osmoreceptors. So again, very commonly asked question, what are the tactile mechanoreceptors? How they are classified? Tactile mechanoreceptors can be classified into, tactile means touch. Mechanoreceptors means what are the mechanical changes that are happening in the receptor. So at the point of pressure, touch, vibration, what is happening in the receptor? Accordingly, changes are happening in the receptor. So you are going to see that these are the changes which are happening. What are the receptors? So remember, encapsulated, which are having capsule, connected tissue. Non-encapsulated, not having the capsule. Encapsulated receptors, mucinous corpuscles in the dermal papilla, Pacinian corpuscles in the dermis, ligaments, joint capsules, Rufinus end organs in dermis and deeper tissues. Whereas non-encapsulated tactile mechanoreceptors are present at free nerve endings in the dermis, ligaments, cornea, bones. Then hair end organs in hairy skin. Merkel discs in non-hairy and hairy skins. So you have to know how the receptors, particularly tactile mechanoreceptors, can be classified. So like encapsulated and non-encapsulated. So then you have to know these are the diagrammatic representation, free nerve ending, expanded tip receptors. You can see the expanded tips, tactile hair receptors, Pacinian corpuscle, encapsulated, then Misner's corpuscle, encapsulated, Cross's capsule, encapsulated, Ruffini's end organ, partially encapsulated, Golgi tendon apparatus, then muscle spindle. So some of the receptors are present in the epidermis, some others are present in the dermis. We will show that also in the next slide. So yes, next slide means after this. So free nerve ending. Free nerve endings are found everywhere in the skin, and in many other tissues which can detect pressure and touch. Merkel's disc, hairy parts, non-hairy parts of the skin contain moderate numbers of these receptors. They transmit initially strong but partially adapting signal, then a continuing weaker signal. Adapts slowly. Therefore, they are responsible for giving steady state signals. Then, Misner's corpuscles, elongated, encapsulated, ending of large myelinated nerve fiber, A beta. Inside the encapsulation are many branching. So you can see the branching. Many branching terminal nerve filaments. These capsules are present in the non-hairy parts of the skin. Misner's corpuscles adapt in a fraction of second after they are stimulated, means rapidly adapting. Then detect low frequency vibration. So then Ruffini's end organs. Multi-branched encapsulated endings. These endings adapt very slowly. Slow adaptation. What is that? We will tell later. They are also found in the joint capsules, help to signal the degree of joint rotation, found in skin and deeper tissues. Then hair end organ. So these receptors adapt readily, and like Misner's corpuscles, detects mainly movements of the object surface on the surface of the body or initial contact with the body. Then Pacinian corpuscle lie both immediately beneath skin and deep in the facial tissues of the body. They are stimulated only by rapid local compression of the tissues, because they adapt in few hundredths of a second, means very, very rapidly adapting. Important for detecting tissue vibration or other rapid changes in the mechanical state of the tissues. Then, many times we ask this, we go dome receptor. Merkel discs, which are often grouped together in a receptor organ called as ego-dome receptor, which projects upward against underside of the epithelium of the skin. So yes, you can see here Misner's corpuscle. This is Misner's corpuscle, rapidly adapting touch and pressure. Then yes, B, we have to know that Merkel's corpuscle, slowly adapting mechanoreceptor, touch and pressure. Then C. So free nerve ending, slowly adapting. Some are nociceptive, some are thermoreceptors, some are mechanoreceptors. Then Pacinian corpuscle, C, D. This is Pacinian corpuscle, rapidly adapting mechanoreceptors, vibration and deep senses, deep pressure. Then E, it is Ruffini's organ, slowly adapting mechanoreceptor in the skin and stretch. So you remember texture of the object, Meissner's corpuscle and Merkel's discs. Localization, Meissner's corpuscle and Merkel's discs. Discrimination, it is Meissner's corpuscle. Vibration sensation, Meissner's corpuscle, Pacinian corpuscle. Prolonged touch, Merkel's discs. Prolonged deep touch and pressure, Ruffini's and organ. Same thing is shown here. You can see. Understood? So then, salient features of the cutaneous mechanoreceptors. So remember, Pacinian corpuscle, onion-like capsule surrounding the unmyelinated nerve ending. Receptive size, receptive field size is large. Adaptation is very rapid, encoded for vibration and tapping. So then Meissner's corpuscle, small, encapsulated, present in non-airy skin, and receptive field is small in size. Adaptation is rapid, and speed of stimulus application is encoded. Merkel's disc, it is a transducer which is available on the epithelial cells. Receptive field is small. Adaptation slow. Encoded for location of stimulus. Then Ruffini's end organ, multi-branched, encapsulated, liquid-filled collagen capsule. So this is the description. Receptive field is large. Adaptation is slow. Encoded for magnitude and duration of stimulus, particularly pressure. Then hair and organs. Hair and its basal nerve. Receptive field small. Adaptation is rapid. Movement of the object on the surface of the body is being encoded. Crosses and organs. This receptive field is small and adaptation is rapid. Touch and pressure are being encoded. So then what are the properties of the receptors? Properties of the receptors: produces generator potential, not the action potential, specificity of the response, graded response, that is generator potential only, then perception and discrimination of the intensity, Muller's law of specific nerve energy, law of projection, adaptation, spatial and temporal summation. What is receptor potential? When a stimulus excites receptor, it changes the potential across the membrane of the receptor. This change in the potential is called as receptor potential. So, when pressure is applied to the Pacinian corpuscle, a small, non-propagated, depolarizing potential develops called as receptor potential. So, when a mild pressure is applied on the corpuscle, mild, non-propagated, depolarizing potential called as generator or receptor potential is being developed. When pressure is increased in steps, the magnitude of receptor potential is increased. Depolarized segment of the unmyelinated nerve ending produces electrotonic depolarization, that is current sink action. I had already discussed under nerve physiology in the first node of Ranvier. When the magnitude of receptor potential is sufficient, above 10 millivolt, an action potential is generated in the first node of Ranvier, which is propagated in the nerve fiber. If still greater pressure is applied on the receptor, frequency of the discharge is proportionately increased. So yes, when the receptor potential raises above the threshold for exciting the action potentials in the nerve fiber attached to the receptor, then action potentials occur. More the receptor potential rises above the threshold level, greater becomes the action potential frequency. More the receptor potential rises above the threshold, greater becomes action potential, yes, frequency. So you have to see that. Understood? Then what is happening? So pressure stimulus resulting in the compression of the Pacinian corpuscle, resulting in the elongation in the change in the Pacinian corpuscle, or deformation of cellular core fiber, opening up of mechanically gated sodium channel, entrance of sodium ions into the core fiber, development of receptor potential, as a result of which development of local circuit of current flow, which is resulting in spread of local circuit of current flow to the first node of Ranvier in the capsule, corpuscle, then opening up of voltage gated sodium channel in the first node of Ranvier, resulting in the entrance of the movement inward of the sodium ions, a generation of action potential in the nerve fiber. This is the mechanism. So yes, different receptors can be excited by in one or several ways to cause the receptor potential. It can be by mechanical deformation of the receptor, which stretches the receptor membrane and opens the ion channels. Number two, it can be by application of the chemical to the membrane, which also opens the ion channels, or by change in the temperature of the membrane, which also alters the permeability of the membrane, or by the effects of electromagnetic radiation. So we can develop receptor potential. So relation between stimulus intensity and receptor potential, amplitude increases and then less rapid rise at high stimulus strength, and frequency of repetitive action potential transmitted from receptor potential receptors also increase, but very strong stimulation action potentials as well, they will decrease the action potential as well. So it is the diagrammatic representation. So then the same thing, so we can see how it is happening, how we can record it. So it is intact capsule, block at first node and cutting the nerve. What is happening at the response? And there is something called as Dale's law. Sometimes we ask this in Viva examination. What is Dale's law? Dale's law states that at one type of synapse, only one type of neurotransmitter is released. Understood? So you are going to tell that this can be for receptor as well as synapse. At one type of synapse, only one type of neurotransmitter is released. Understood? So what are the properties of the receptor potential? I had already mentioned receptor potential is not action potential. It is similar to excitatory postsynaptic membrane potential, EPSP, or end plate potential, EPP, in a neuromuscular junction, or electrotonic potential in a nerve fiber. There are important characteristics. Graded response. What is graded response? It is the receptor potential, which is supposed to be a graded response. It is meaning that its amplitude increases with increase in the velocity of the stimulus application, increase in the strength of stimulus, increase in the velocity of the stimulation, as well as increase in the strength of the stimulus, automatically increase the amplitude of the response. Then number two, it is showing summation. Receptor potential from two stimuli can be added if second stimulus arrives before receptor potential developed due to first stimulus is over. before it is over. If the response of the first stimulus is not at over, if the second stimulus is applied, response is being added upon. Thus, receptor potential, unlike action potential, which cannot be added, receptor potential can be added together. Summation. Then refractory period. Refractory period is not there in the development of receptor potential, while action potential has refractory period of 1 millisecond. So I will talk about this in a tabular form. So refractory period of 1 millisecond is available with your action potential. Understood? Then local response, receptor potential cannot be propagated. Duration of receptor potential is greater. It is about 5 to 10 milliseconds, whereas action potential duration is about 1 to 2 milliseconds. So we know receptor potential is a graded potential. It is non-propagating. It does not follow all or none law. It can be provided in the comparison table like this. So what is that? Generator potential is a localized one. It is action potential is a propagating. Then magnitude of generator potential decreases with the time and space, whereas magnitude of action potential remains same, and generator potential is a graded response. Action potential, it is following all or none law. Generator potential lasts for 1 to 2 milliseconds, whereas action potential lasts for 0.1 to 0.2 millisecond. Generator potential has no refractory period. Action potential has refractory period. And generator potential has got tendency of being summated. It can be summated. Action potential, no phenomenon of summation. So what is specificity of response? So yes, next characteristic is the specificity of the response. That is, specificity of the response or Muller's law refers to the response given by a particular receptor to a particular sensation. For example, pain receptors give response to pain sensation. Temperature receptors give response to temperature sensation. Each type of sensation depends upon part of the brain in which its fibers terminate, where the fibers of the concerned afferent nerve arising from concerned receptor terminating, that is going to decide. Specificity of response is also called as Muller's doctrine of specific nerve energy. So remember, this is something called as an interrelated topic. So you have to know that specificity of response when you are going to stimulate, yes, Pacinian corpuscle, touch sensation, pressure sensation, Ruffini's receptors stimulated, temperature sensation. Understood? So you have to know that is being produced. So what is Weber-Fechner's law? Intensity discrimination. So if the strength of the stimulus, intensity is about 10 units, an arbitrary unit, sense of stimulus pursued, strength of stimulus pursued is log of 10 to the power of 1, that is 1. If the strength of the stimulus is increased to 10 times, that is 10 to the power of 2, 100, perception of intensity is not increased by tenfold, it is increased by 2 times. So that is because log of 10 to the power of 2, it is 2. If the strength of the stimulus is increased to 100 times, that is 100. Yes, what is that? 1000 unit, arbitrary unit being applied receptor, how much is the perception? Perception intensity, strength of reaction will increase to 3 times because log of 10 to the power of 3, it is 3. Mathematically, so our reaction or sense perceived is equal to alpha is a constant, log of, yes, it is strength of stimulus. So R is the intensity of the reaction or sense of perception, alpha is the constant, S is the intensity of the stimulus. It is advantageous. In reality, the pain is more actually, but what is happening in our system? Pain will be perceived less. It will be perceived less. So yes, what is sensory coding? So you have to know that there is coding of sensory information. Receptors encode 4 elements of the stimuli. Modality, what type of modality? What is the location? What is the intensity? How much is the duration? So there is a differential sensitivity of the receptor for a particular stimulus or specific energy for weight. It is designed. When the nerve fiber from the receptor is stimulated, the perception is that for which the receptor is specialized, no matter where and how that nerve is stimulated. That is called as Muller's law of specific nerve energy. Specificity of nerve fibers for transmitting only one modality of sensation is called as labeled line principle. Later on, I will highlight about this also. So, coding of sensory information, moderately crude touch is applied on the part of the skin. Even with the closed eyes, person can feel it as a crude touch. It is just crude touch, not very crude touch. Person can localize the site of touch sensation from where the sensation is arising. By these characteristics, person can characterize any conscious sensation. Like it's what is the type of sensation that is generated, where exactly it is being pursued, how exactly it is, how long it is being pursued. Each nerve tract terminates at a specific point in the central nervous system. Type of sensation felt when the nerve fiber is stimulated is determined by point in the nervous system to which the fibers leads. For instance, if the pain fiber is stimulated, person pursues pain regardless of what type of stimulus excites the fibers. Likewise, if the touch fiber is stimulated by the electrical excitation of the touch receptor or any other way, person perceives touch. Understood? So this is called as coding of sensory information. What is law of projection? So law of projection states that no matter which part of a pathway you stimulate between the receptor and the brain center, your brain will locate the stimulus where the receptors are. So reorganization of the brain map in the somatosensory cortex is available. Some believe it can be blocked by local anesthetics in the spinal cord. So it states that sensation is localized at the site where receptor is located, even when the stimulus is applied at any point on the sensory pathway. So what is phantom limb phenomenon? Phantom limb is a sensation that an amputated or missing limb is still attached to the body. Approximately 60 to 80% of the individuals with an amputation experience phantom sensations in their amputated limb, and majority of the sensations are painful. Phantom sensations may also occur after the removal of body parts, such as after amputation of the breast, after extraction of the tooth, phantom tooth pain, after removal of the eye, phantom eye syndrome. Then, what is adaptation? Adaptation, when the receptor is continuously stimulated with the same strength of the stimulus. When the receptor is continuously stimulated with the same strength of stimulus, after some time, the receptor stops sensing impulse. through the afferent nerve. It is called as adaptation. It is dependent upon, yes, there are, basing on this, there are two types of receptors. Another way of classifying, basing on the adaptation. Phasic receptor, rapidly adapting. Tonic receptor, slowly adapting. Phasic receptors, which adapt rapidly, touch and pressure receptors. Tonic receptors, which adapt slowly, pain and temperature, chemical, chemoreceptors and baroreceptors. So this is the way, fast adapting or phasic. So yes, phasic, tactile receptor, touch receptor, olfactory receptors. We should not forget this. Understood? So then slowly adapting, tonic receptors, proprioceptors. Yes, non-adapting. Actually, non-adapting. This pain is non-adapting. Baroreceptors, non-adapting. Understood? So yes. So adaptation of the receptor, another characteristic of all the sensory receptors is that they adapt either partially or completely to any constant stimulus after a period of time. That is, when a continuous sensory stimulus is applied, sensory receptor responds at a high impulse rate in the beginning and later at a progressive slower rate until finally rate of action potential decreases to very few or often to none at all. Information carried to the higher center becomes zero sometime. So that conservation of energy, conservation of neurotransmitter, all these things can happen. Then mechanisms by which receptors adapt. Part of adaptation results from the readjustments in the structure of the receptors itself. Part from the electrical type of accommodation in the terminal nerve fibril. Slowly adapting. What are the slowly adapting? Which are the rapidly adapting? Slowly adapting impulses from the muscle springs, Golgi tendon apparatus to the nervous system to know the status of the muscle contraction. They are slowly adapting. Then receptors of macula in the vestibular apparatus, pain receptors, baroreceptors of the arterial tree, chemoreceptors of carotid and aortic bodies. Then rapidly adapting are the phasic receptors, Pacinian and Meissner's corpuscles. So they are the examples and how. So yes, what is summation? So summation, we have already mentioned, summation of the stimuli applied simultaneously on the receptor or of stimuli applied repeatedly at the same site. So stimuli applied at the same site. So yes, same site, that is called as temporal summation. One after the other. So stimuli are provided on the receptors. Strength of the stimuli are subthreshold, as a result of which effect is being added. Stimulus number 1, stimulus number 2, and summated information you are going to get. So here, temporal summation. When the stimuli are applied one after the other in quick succession, and they are of subthreshold intensities, as a result of which you are going to see a temporal summation. Then you are going to have stimulus number 1, small, yes, subthreshold, and somewhat better is the second stimulus, subthreshold also. It is also subthreshold, and when that is provided before this particular effect is over, you are going to have summation, that is called as spatial summation. Kindly recall from my discussion regarding synapse, spatial summation and temporal summation. This will be the last property under receptor I am going to discuss. What is being referred to as lateral inhibition. Lateral inhibition. It is a phenomenon, important property of the sensory receptor, helping in the sharpening of the margins of the stimulus, as a result of which one can make out the actual position of the object. Say, for example, an object is being placed. This is object X. It is stimulating receptors A, B, C, and you are going to have receptive fields small a, small b, small c. Understood? It is in the receptive fields of 3 receptors. Understood? And you are going to stimulate these receptors, and potentials are developing in these 3 receptors. In the sensory pathway, when first order neuron is synapsing with second order neuron. First order neuron, second order neuron. First order neuron, second order neuron. First order neuron, second order neuron. 3 pathways. When in sensory pathway, when first order neuron synapses with second order neuron, former also inhibits adjacent neurons of second order. So you are going to have interneurons. So this B is bearing a collateral. This collateral is in communication with a neuron, which is an inhibitory neuron, synapsing onto second order neuron of adjacent pathways. Second order neurons of adjacent pathways. As a result of which, as it is by nature inhibitory, signals further not carried in the pathway A and C properly. Magnitude of information conveyed through the second order, third order neurons will be progressively reduced because of this interneuron as a result of collaterals. These interneurons, which are by nature inhibitory. Understood? So you are going to have, when B is stimulated more and more, A and C will be stimulated more and more. A and C. Activity of second order neuron of B will be more. As a result of which, you are going to see second order neuron activity in the pathway A and C will be less, so that individual is able to identify the exact location on the surface of the scale. The object X is being placed on the surface of the scale. Its exact location can be made out. Lateral inhibition is a phenomenon associated with the receptor, by virtue of which individual is able to identify the exact location of the object. So here another diagram I had depicted. This diagram, it is for 2-point discrimination. So yes, this is over the back. 2-point discrimination, the receptor density will be less, whereas at the tip of the tongue, the density will be more. 2 points of compass astiometer. These 2 points which are applied simultaneously can be identified as 2 different stimuli. Distance is very small, less than 1 millimeter, even 0 at the tip of the lip, tip of the tongue. Understood? It is called as density of the receptor. So density of the receptor and lateral inhibition are being useful in sharpening the edges of the receptor, sharpening the edges of the margins of the receptors. Understood? This is going to complete the topic. For MSc students, you must be knowing by this time. So you are going to have second IA examination, descriptive type. You have to write the answer sheet and you have to upload the answer sheet. That will be on 27, 28, 29, between 11 to 12 for MSc students who are attending along with MBBS students here. Information 27, 28, 29, between 11 to 12. You will be having anatomy, physiology, and biochemistry second IA examination for MSc students. So this is going to complete this topic. In the next class, I will talk about reflex. You must be following now. Yes, sir.
Yes, sir.
So multiple slides I had included so that you can have revision, you can have reinforcement of the information. Understood? ಎಷ್ಟು ಜನ ಇದ್ದಾರೆ ಹುಡುಗರು?